Hello everyone, welcome to Get My Class. So let's solve this question here. It says in Meselson and Stahl experiment, cesium chloride is not used for option A, detection of radioactive isotope. Option B, helps in measuring the density of the DNA. Option C, density-based separation. Option D, both A and B. So let us understand the question. It says in Meselson and Stahl's experiment, cesium chloride is not used for. So they're asking like cesium chloride was not used for which among the following when Meselson and Stahl's experiment was going on. Now, when we talk about Meselson and Stahl, what comes to your mind? Of course, they are the people who proposed the semi-conservative mode of replication, right? So Meselson and Stahl's experiment proved that the DNA replication occurs by the mode called as, or a method called as semi-conservative method. Semi-conservative method, right? Or semi-conservative mode. Now, during this experiment, they have used cesium chloride. What did they use? They used cesium chloride. Okay, the cesium chloride helped them in identifying, or you could say, helped them in separation of the DNA strand, which they, where the cells were grown on N14 and N15 media. Okay, individually. Okay, so that helped them in separating the various types of double-stranded DNA. Okay, like it helped in the separation of N14, where both the strands of the double-stranded DNA consist of N14, and it also helped in the separation of, you can say, N14, N15, which formed a band, which is a hybrid band. Okay, why? Because one of the strand of the double-strand DNA will be containing N14, whereas the other contain N15. And finally, they also saw the band where it had N15, N15, okay, where both the strands consist of N15, N15. But this hybrid band was always there. So that means that the parental strand has been conserved. Okay, so that is how they prove the semi-conservative mode of replication. Now, cesium chloride was used as a density gradient to create density gradient. Density gradient. Okay, so during centrifugation, what happens? Like based on the density, the double helical, or you could say the double-stranded DNA will get separated based on whether it consists of N14, N14 or N14, N15 or N15, N15. Okay, so that's how it creates a density gradient. That is, cesium chloride creates a density gradient. And what is the reason for that? It helps in the separation of these double strands of DNA containing N14 or N15 or the hybrid of both. Okay, so let us see. Option A says detection of radioactive isotope. No, it was not used for detection of radioactive isotopes. Because you cannot, by using cesium chloride density gradient, you cannot detect whether it is radioisotope or not, okay, or radioactive or not. Hence, it is ruled out. Okay, but then since they're asking about not used for, then this is the correct answer because cesium chloride is not used for the detection of radioactive isotope. Okay, so though the description is wrong, but then we are looking for the wrong statement. So hence that is the correct answer. Option B says helps in measuring the density of the DNA. Yes. So you will know whether it is a hybrid band or pure N14 band or pure N15 band. So it definitely helps in measuring the density of the DNA. Then option C says density-based separation. So as I told you, cesium chloride creates a density gradient, which helps in the separation of the double-strand DNA based on its density. So that is also correct. Option D says both A and B, which is not our answer because we are looking for the one which says like in Meselson and Stahl's experiment, cesium chloride is not used for, it is not used for detection of radioactive isotope. So that should be the correct answer for our question. So let us conclude. So the answer is option A. Okay, I hope you understood the explanation. Thank you.